warm welcome to our respected chairman, principal, interactions, chief guests, dean, HOD, teachers, and my dear friends. I, Vanuja, feel a great pleasure to welcome all of you on this afternoon for an expert talk on NextGen jobs and entrepreneurial skills organized by Institution University Council, IISc. On behalf of Department of Biotechnology, the Oxford College of Engineering, I take this opportunity to welcome our esteemed chief guests, Dr. Rajnikant Vaidana, Dr. Sumati, and Dr. Dr. Rajnikant Vaidana is a founder of New Technology Strategy,
right? You cannot stay static in 1970s and say that no, kids have to come here, they have to learn this, they have to do this. That's not what it's about. According to your syllabus are changing, according to your mindset is changing, your access to information is so easy. Tell you does it have nobody. Everyone. So there was the time when I used to go into bioinformatics at room temperature, ambient temperature for 10 to 15 years. And you can do all things, anything and everything, all sorts of things. That was an outcome of the realization, where a real problem was taken and a solution was developed with the mindset of helping the society. Now this project is being supported by incomes from utilities, Department of Biotechnology, and just 2 months back, the Government of India and Department of Biotechnology has officially launched this project into the market. What internship started in 2016 and 17, sometime back, 10 years back, has resulted in a product now. It takes that much of hard work to come to this stage, which is incredible. Right? It's a real thing. But once you build a solution, without even your knowledge, it will extrapolate itself, because one fine day this solution was taken by Dr. Gini to a place called Kalkinos Diagnostics in Cochin. So one of the pathologists, he put the tissue in it and it is histopathology, and it was better than normal fixative. You know what is the fixative which is used in histopathology? Formaldehyde. It's a very toxic potent carcinogen with a very bad antifungal solution. But we have no alternative. But suddenly, we got an alternative. So it's not just for sample preservation and collection, now it has become an important solution for tissue fixation. And it is 100% advanced. Completely not. Suddenly from nowhere, we are in competition with formaldehyde. And then we went to some of the conferences, and one of the medical doctors suggested this can be used in anatomy. I can replace all the anatomy samples, plant samples, plant research institutes. Zoological Society of India is putting its specimens into this, and anatomy labs are changing from formaldehyde to this solution. And lastly, the most interesting thing is now cadaver embalming. Not only passes away in long distance, they are complete embalming. And that embalming is done with formaldehyde. Right? And when such a body goes into the earth, anything being put to creation, it's a very bad solution. Right? This replaces that. So without even our own knowledge, without even our own intentions of getting into, the product has made its way into several things. The technology is spread by itself. Now we have another part of it wherein you can store your cell lines, you can store your microbes, all at room temperature. You don't need frozen. So things started to evolve themselves completely without even our own knowledge. And that's what an innovation does to you. An innovation does to you when you have the right team. Right? So the team and innovation go hand in hand. So innovator needs to have a good innovation team. Right? And when COVID started in 2020, we just registered the company and we came up with an idea that we need to be able to detect in the airport or in the bus stops or railway stations within 15 seconds.
About how the industries work in biotech or let's say life science. What do you think is the theme of the talk? How it's going to be? Yeah, yes. How? How we are solving the real yeah, number one. And yeah, and so one thing which was very dynamically presented was that we are not just a life science company. The theme which we work on is there is a lot of departments which work together in vision of that particular particular goal, particular product. Me coming from electronics or IT background, working in this current field for almost seven and a half years, worked with India government, worked with almost 30 startups. Everybody, if you be very constrained, I don't think there is a future in MNC or a startup, which is a real problem right now, which people face. A lot of youngsters have been co-guides for a lot of projects like in BMS and in them, and many students come and meet me. The only problem is that they'll be very focused. They come from one stream and they're like, no, the topic has to be that. Please don't, because you might do it. You might love it when you're doing it when you're in college. But when you go and search for a job, then that makes you a very cornered person. Because that also showcases that you're not okay to work in collaboration with many other things. You might not be comfortable, you might not be easygoing in working in collaboration and interfacing a lot of things. So please keep that thought in mind, that the main origin of the talk for today, that we all three of us come from a very distinct field. She's from genomics background, I'm completely microelectronics and mechanical background, he's from physics and all clinical background, but we all have that say in the product. There is a procedure which we follow in order when you look for it as a product. And any technical job if you're looking for, nobody will give you a SOP. SOPs only work till you're in college. So there will be no experiments given in order that, okay, somebody will come and tell you that next step should be this. So you should be proactive. That, okay, if I have this vision like she was talking about, I have a vision. So you should also be very proactive in that, okay, if that is the vision, what step I can bring in? Because that's very short, like we all come from very different generations, like sir told. You might have some new idea which we might not be knowing, and an easy way to solve it. So why not? So don't be scared of trying or putting your opinion or trying out something new. And any journey, you work in a Siemens to GE to Biocon to a startup also, or you work with I work with incubation centers also, a very small level startups to big level startups also who got graduated, got funding, everything. It's a process. You cannot get anything, just in a flick of a second. That you pass your college, I meet students and they're like, okay, I have to get, this is my benchmark. This should be my salary, otherwise I will not do it. No, this is not how the industry works. Why they should also trust you. You have to do something, right, to create that, or prove, not just prove, you have to build that trust with certain organization. So it's a process. In process, you have to have a vision, you have to have a goal, and you have to be very flexible. Extremely flexible. That today, if I got a degree, I should not be hesitant of doing something smaller. No, because end of the day, you are contributing in something. Me coming from a biomedical or completely mechanical, electrical background, how do you think I'm attached to these products? Because we brought it to this level. After this, how will you maintain it? A chemical solution has been designed. Now, samples have been stored. Now, everybody liked that, okay, for 10-15 years I will store the sample. What is the end use of that sample? At the end of the day, anybody, any medical or a clinical background would like to have the data. Your asset is your data, not that organ which is inside. So, at the end of the day, all this NGS, DNA, RNA, protein, what analysis you'll be doing has to be stored. So, it's not just storing the organ or any sample for 10 to 15 years. It's about storing a data also for 10 to 15 years. Like few years back, you got COVID. People ran to similar diseases in order to build the vaccine. But what will happen if something new comes up? People will look around like somebody who has any data. Then you come in picture. Because you're not just having an organ. At that crucial stage, nobody would like to split a lungs and see what is happening. Right? But people would like to go back in search for the data. When you start developing something, you go and do your project. What do? You go for literature survey. What is literature survey? You do a history analysis. This is just the same way. So we come in picture. They did their part. But our job is they created a biobanking solution basically. You can store something for longer time. But we will also be helping them in building the archive. We are the one who will be building backstage that virtual entity who anybody might need. So everything works hand in hand. Now talking about the device. So this device particularly, I will not talk about only about this device. It's a platform. Like you heard about InstaPreserve. This is InstaDetect. It's a chain umbrella which is InstaDetect. Now what is InstaDetect? InstaDetect is not something which builds point of care small diagnostic devices. No, InstaDetect is a platform which uses proteomics also in order to build small peptides to improve sensitivity, specificity. We make our own gold nanoparticle, iron nanoparticle, silica nanoparticle also we were making to enhance more and more about the sensitivity and how fast you can generate something, how fast you can get a result by improving it more and more. So nanotechnology is also involved, proteomics is also involved. Then comes now the science part is taken care of. I know how my assay will be built, which is a small LFA, let's say in the heart device one. Okay? When what we was talking about the breath-based COVID device when we made, we were making a rapid test kit using the saliva because that time we wanted to use an invasive method because it was very uneasy for people, for kids to getting swabbed at. So the first version of that product was a simple rapid test which was done using a saliva. But then when we saw the data of that rapid test kit, we understood, okay, there is a change which we can see on this strip, right? Why can't we make it in a bigger block of like a square sheet? And no coating, no nothing. Let it change the color and the specification, optical density, what all getting changed. And we will record it. And then that's how, from a rapid test kit, we moved the similar technology, we moved towards developing a small device, which becomes easy for people to keep it anywhere. You can ask even a kid also to, breathe out in that and it will absorb and give you the data. The similar way, this also is made. So it has the particular peptide for the specific biomarker, let's say for troponin. Then we have also in-house made, we have a nanotech team which develops all sort of nanoparticles as small as 5 nanometer to as big as 60. So the COVID, which I'm talking about, used 8 nanometer nanoparticle. Hard to extract, we failed a lot of times, a lot of time because one lot we made, right? There are 5 people. The other one made, maybe it became 5.5 nanometer. 8 I needed, it became 10. 8.2 also will create a cost to me. I cannot get the duplicate data of that. And I cannot build in the device. So it's a procedure. The same way, this also involved nanoparticle, this also involved peptides, this involved a rapid test kit like team, which are pro in making the LFAs, pro making rapid test kits. And once this was done, then we made a device. So what this device particularly does, it's completely on your image sensing. It will capture all the images at the particular millisecond based shutter speed, and what will it do? It will combine it and then it will calculate. So what will do in the calculation, what will happen that once the image has been recollapsed and everything is done, then it will read is there any intensity difference because it's a quantitative device. So there will be slight change in the intensity in the current LFA, the device will detect by doing the image analysis. And to do that image analysis, because we had a lot of data, so we used AI. There is no machine learning. We have to automate it because every time we cannot sit in the emergency ward. The same with the COVID. We cannot sit in the COVID ward, although we did that time because there were very less people and you can say there is no other option. We were so passionate to build it that the entire team of 8 people got COVID for not less than 10 to 11 times because we were sitting when we were getting infected, while people were resting, we were sitting in the lab collecting our own data. because from where you get the data? You have to train the device in order to become it more sensitive. Machine learning is very, fascinating word. Everybody would like to, machine learning everywhere I go. Even a physiotherapy college is doing machine learning. I don't know how they combine it and use it. Very fascinating. But how you use that machine learning? You need tons and tons and tons of data to generate that data. Every time a patient will not know, I am asking him to breathe out, he's sitting on my hand, I got COVID, done. Right? So you get infected, you're still working in the lab. We were holding tables and walking and collecting breath samples and doing it. So, but we all were so passionate with a team of 8 people. No matter if you're getting COVID, you're sitting and making the device. So there was no excuse because we were asked that if we want to go home, we would have easily been in our home at rest. But it was we were passionate. It's okay. We all were suffering together. Nothing okay. We all were suffering together. We locked the lab. Nobody should go out. Nobody should come in. So it's a process. The same way to build this InstaDetect platform, there are a lot of teams I have to work with. So I cannot keep this, taboo in my mind. Or I cannot keep even more, I cannot show them cornered that, okay, I'm the one who will be giving the share of the device. You cannot sell sometimes I joke to them, okay, that you cannot sell your science. I have to give it a product look, then only you can do it. But if they will not give me science, what product I can make? I need it. So it's a hand-in-hand job, right? So this is about the platform and how the platform have been made, how different fields have been involved to build this particular platform. The similar way in this platform where we come in. So we all work together. And that's the main motto if we talk about the way we provide trainings, the way new youngsters, kids who do project with us. We keep this, that you have to keep switching, keep working with different departments. What if I made it today? And tomorrow a person who might have a gastric pain getting it tested, but I have not got it regulatory approved. It is not journal electrical safety approved. The person who's testing might die with the shock because there are batteries inside. We will be using liquid inside. What if it blasts? So regulation is highly important. I need it.
So the product name now what we have labeled it as commons to preserve. So where you can store. I'm sure that you know how samples are collected, how is it transported. Most of them use cold chain system, either the liquid nitrogen or -20, -80, which is very tedious. I don't know how much of you have real experience of handling clinical samples or any other samples. Sorry? Yes, toil. And during toil also, you lose the sample viability. We think that it stores the sample, but as 2 or 3 times before the sample viability is actually lost. We don't consider all those, and also there is a cost involved whenever you collect and transport the samples. There is a lot of logistics involved in the collection, transport and things. So that is why there was a need actually. There was really a need to identify some solution which can be used to collect the samples at room temperature or at ambient temperature. And that is why we developed this solution. This was not this actual how it looks like. It is not how it looked like. There's different prototypes which was initially, it looked more like a gel actually when it was developed because the composition was different. But then we modified it. Now we have, it's not a one SKU or one product. I would say that now we have 10 to 11 products. The base remains still the same base of ingredients that is required for it remains the same, but we have developed over it to come up with different solutions which can cater to different fields actually. It is not just molecular. Initially we thought we can use it only for the molecular work for collection of blood and saliva and use it for the downstream and not only just collect the samples, it should also retain the integrity of the samples so that it can be used for the downstream. Right? What after collection? So you need to have the DNA, I'm sure, right? What is the downstream that you would really look for? Any idea? DNA, RNA, proteins, and biomarker, definitely. This is all biomarkers. Either it is you look at the genetic marker or you look at the protein marker, right? Which will help you for the diagnosis. Everybody would be always heard of this NGS, which is a very hot term now, right? So we have also validated using our NGS, which is the high throughput system. not only real-time assay where you quantify the gene expression patterns to identify the genetic cause of the differential marker patterns, but also in the NGS system. We have our own NGS system and we have validated that. That is how, and we have got a BIRAT grant for this. So that is how it started from there. And then we have developed and we started. So the second step for any product is then going to the market. How is your market accepting this product? Right? So that's the time when we identified that, there are so many things which we may not really look at it because we have a closed mind, because as when we develop, we think that only this is possible. But then our customers told us, why don't we try this? That is how our product have really branched out from only molecular work to histopathology work, to anatomy work, to embalming. So now our solution is even being used for the embalming, which is, I think, which is really great achievement, I would say. And very recently we had attended a conference on anatomy, and Dr. Meenakshi have actually, she's Amazon Hospital's HOD, where she has established this, and she told that when we use formaldehyde, what happens for the clinical samples or for the fetal samples, it's very rigid. There's no movement. So when the students, medical students, have to really dissect it, it becomes very difficult. And plus, Oxford Medical College, this is one of the experience that the students of Oxford Medical students have shared with us during that conference. They're so frustrated using formaldehyde because of the smell it has. Not the smell, because of the irritations. When we go out to the anatomy, you would have really seen your eyes starts watering, your respiration system is really affected, and they're forced to, 4 to 5 years, they're forced to work in that environment. So I think such solution can really revolutionize how, without affecting your work, but not affecting your health. So they're compromising on health at certain levels, not only the students but also the pathologists. So we target that it should really be economical and plus help them to work in a very friendly environment without hampering your health system. Because pathologists are doing a lot of work actually. They really, especially when the cancer diagnostic comes. We know the cancer currently diagnosed is mainly done only by the histopathology work. Genomics has certain aspect to it.
So you need to just select the option, OK profile. Just keep on pressing next like terms and condition. It will take the results to you. And it actually, it's a 15 minutes process. So in 15 minutes, every milliseconds, it's capturing. So there is a camera inside. It has a very highly sensitive camera which has a shutter speed. Okay, so the shutter speed has been modulated, it's coded.